So our next presentation is going to be given by Suresh and Birkholz. They're going to talk to us about Cyst stem cells.
Hi, I'm Birkholz. This is Suresh, and we study the function of the BMP dpp in Cyst stem cells.
Adult stem cells are undifferentiated cells that replenish our bodies with cells. They have amazing capabilities in the field of medicine because of their ability to repair damaged tissue. We use the Drosophila testes as our model because it is a good standard model, it is easy to obtain, and it is easily genetically modified. Here is an example of the end of the testes with the hub cells, which are a group of non-dividable stromal cells with germline stem cells surrounding that, and then cyst stem cells surrounding germline stem cells. So the hub gives off self-renewing signals to these stem cells, which allows for asymmetrical division. Asymmetrical division is when one daughter cell stays a stem cell while the other differentiates, in this case, the spermatogenesis and becomes sperm.
Okay. One of the types of signaling used from the hub to the germline stem cells and the hub to the cyst stem cells is called the signal transduction pathway. And basically what happens here is the hub emits these types of proteins called ligands, and we study BMP and dpp. And these ligands basically up here, they go into the receptor on a certain type of receptor that's specific to a germline stem cell or a cyst stem cell. And when this connection is made, the proteins are activated within the cytosol, and once these proteins are activated, they're then able to enter the nucleus of the cell and make changes to the gene expression. So by either turning genes on and off, so that the cell knows when to differentiate and what to differentiate into. Here we see that our hub is producing these bone morphogenesis proteins, and that's just a type of signal transduction pathway. So when these BMPs are emitted by the hub, which are both the ligands GBD and dpp, the germline stem cell gets a signal to self-renew. So the germline stem cell knows that when it's going to stay a stem cell when that's close to the hub, and we're going to differentiate along spermatogenesis. So when in previous studies, when the signal transduction was taken away from the whole stem cell leaf, all of the germline stem cells differentiated and there were no stem cells left, which makes sense. But when they introduced the signal transduction back into the cyst stem cells, these cells were actually able to do something that told these germline stem cells to continue self-renewing again. So we know that the cyst stem cells emit BMP-dpp. So we're thinking the other signal that is necessary for self-renewal could possibly be dpp. So that's what we're studying. Here we have a diagram of a section of DNA, and basically on your left is what's most important, these upstream enhancers. And these enhancers are basically just, to us, the random sequences of DNA, and what they have the potential to do is turn the genes found downstream on and off. So what we did is we found this DPP gene, and we're going to take random sequences of DNA, and we're going to test them to see if they're enhancers by turning genes on and off. And the way we do this is we put them into the recorder gene. And what happens here is we have our potential enhancer, and we put it in front of a protein called GAL4, and then that's in the female side. And then in the male side, we have the recorder gene that has this orange ball for a UAS receptor, and they go in front of a protein called green fluorescent protein. So when these two slides are crossed, if this enhancer is able to turn on the gene, then GAL4 will be produced, GAL4 will bind to the UAS, and then when this connection is made, GSP will be produced. So that's how we know the enhancer was actually able to turn on GAL4. And GSP will be able to see in our microscope slides later. So our main goal for this experimentation was to determine whether we had found the DPP gene enhancer by putting it as a substitute, like GAL4 is a substitute for DPP, so we see if it can enhance GAL4 instead. And then we test it using GSP so we can see it by fluorescent staining, which is next. And then if we see GSP produced in the cyst stem cell, then that means that DPP is being produced in the cyst stem cell.
Immunofluorescence staining is the method we use to find our gene of interest. Here we have the protein of interest specific to our hub, germline stem cell, or cyst stem cell, and our primary antibody attaches to our protein of interest, and then our secondary antibody attaches to our primary antibody. Our secondary antibody has fluorescence, which is what we see when we look at it under the microscope. For our method, we start out by copying lines with our gene of interest and then dissect the protein. We do a coarse dissection where we take out the end of the flag with the organs and testes intact. Then we add formaldehyde to poke holes in the cells so that our antibody can get into the cells. And then we wash with TBTX to get rid of extra formaldehyde so that it doesn't poke too many holes. Then we add blocking solution, which helps protect our other proteins not of interest from our antibodies so that we don't have extra noise when looking at it under a microscope. Then we add our primary antibodies, which are formed in the animals on the left, against the proteins on the right, which are GFP, which is green fluorescent protein. That is what we are looking for. FASA is germline stem cells. Class 3 is hub cells, and PJ is cyst stem cells. And then we wash with TBTX to get rid of extra primary antibodies. Then we add our secondary antibodies, which are all made endogenously. And these are the ones with fluorescence, which we see under the microscope. And their colors are 488, which is green, Cy 3 is magenta, and Cy 5 is white. Then we wash with TBTX to get rid of extra secondary antibodies. Then we add glycerol and ringer solution. The glycerol helps keep the testes stable while the ringers help preserve the testes. Then we do a fine dissection where we remove the testes individually and place them carefully on a microscope slide where we look at it under a microscope and we look for GFP.
And we have our results. And here's our first testes, and what you're looking at basically is the very end of the testes right here. And if you remember in the first slide, it was kind of a curled shape, so this is the very equal end. And the way we know that we have the right place is on the right here, you'll see that the hub is colored in magenta, and if you see the hub, then you're seeing the stem cell niche. So you see the hub here. And if you notice in all these pictures, it's all the same picture. It's just that some of the colors were taken away so you can see what's under the right colors. And we see a lot of green, but it kind of seems scattered randomly, kind of like background noise. So what we're looking for is more focused grouping with GFP. And if you notice here, there is a less specificity going on. And over here we have our cyst stem cells in white for our focus. And these cyst stem cells don't really correspond to any concentrations of GFP in the middle picture. So if we had a positive result, a very clearly positive result, then we would see wherever these white parts were, then we would see like concentrated green spots. But we didn't. So we determined that the result was negative. Here we have our second tested, and we see that we have a kind of like more concentrated grouping of GFP, more localized. And on the right, we have our hub in magenta, and we see that this corresponds to this picture here. So we know that the hub is producing GFP, and that's what's expected because we know that the hub is able to produce GFP. So on the left here, we have our nuclear stem cells and our cyst cells, and we have the cyst stem cells don't really seem, again, to be making much of a difference in any concentrations of GFP. So we determine that the result is negative. Here we have our third tested, and we have even more clear grouping of GFP. So that kind of gives you a better idea of what it would look like when something is producing GFP. And in this case, we know that it's the hub because these pictures correspond to each other. And on the left, we have our cyst stem cells in white again, but here we see that, like, for instance, this one doesn't really seem to be doing anything over here, and we determined that we weren't producing with these very high levels of GSP. So we determined they were negative. Here we have our force tests, and these are all different genetic, like different gene enhancers. So each of these testes was used with a different sequence of DNA as a possible enhancer. So here we have our force tests, and we see that we have another more solid grouping of GFP, which corresponds to our magenta hub over here. And then on the left, we have our white cyst stem cells. And again, there isn't much difference. You don't see a big green blob here and a big green blob there. So we determined that the result was again negative. Here we have our cyst tested. And if you notice in this picture here in figure C, there's a lot of magenta, like a lot more than we had in the other pictures. And this is mainly because our VASA antibodies for the germline stem cells and the nerve cells, as we go farther up, it didn't really bind very well to the proteins in the first four testes, and this isn't really a problem. It just means that we could see the germline stem cells before. So now we see them, and we know that they're there. Nice. And we have our concentration of GFP, which is very clear in the testes. So the hub is definitely making a lot of GFP, very concentrated. And we have our stem cells on this side again, who's white, and we don't really see any correlation between the two pictures, so we determine that the result is negative. So just a reminder, we have our gene enhancers, which were found upstream of BTP, and we didn't know if they were enhancers yet, so they're basically just random sequences to us. And we put in that certain DNA sequence in front of a protein called GAL4, and then GAL4 and UAS would bind if it was produced. And once that bond was made, then GSP would be produced. And GSP and a progeny would be seen in these immunofluorescent stains. And for all of our testes, except for really the first one, we really saw production of GSP in the hub, which was expected. But then something interesting was that in the cyst stem cells, we didn't really see clear production of GSP, like large amounts. But if you notice in this picture, it's very clear that we have what we call a web effect kind of looking. And if you remember how the stem cell niche works, is the hub sends out signals, and when one stem cell divides, it no longer gets the signal. So if you have to differentiate, you get bigger and move down the testis in this direction. So these kind of darker spots are the germ cells, which have now begun differentiation. And around it, the cyst stem cells, when they differentiate, they kind of wrap around. And if GSP was being produced in the cytoplasm of the cyst stem cell, then it would kind of make that wrapping shape, and you would see a picture that kind of looks like this. So that made us believe that we might be seeing cytoplasmic staining of the GSP for the cyst stem cell. Now, we expected GSP to be produced in the nucleus of the cyst stem cell, which is why we stained the nucleus of the cyst stem cell. But we would have to do more analysis on these images and zoom in and take away the white layer to see if we can see a very faint or like webbed effect over the stem cells to see if there was GSP in the cytoplasm. So there'd be more analysis on these images needed to see if our negative results are actually accurate, or if it's just a lot of background staining. Okay. I would like to thank the 1981 alumni for sponsoring my stay here at SI. Without her, it wouldn't be possible for me to be here and have these great experiences and opportunities. And I'd also like to thank the Kinder Morgan Foundation and for their funding my research and making it possible for me to work with Dr. Levinan and Tim Hammer in their lab and do this amazing research at the Big City.
I'd like to thank the Morris family for funding my research and my stay here, and we'd like to thank Judy Leatherman and Ken Hammer for mentoring us and teaching us about the stem cell niche and letting us do this awesome project.
We'd also like to thank Tony Gundana for being our awesome advisor and editing all our papers and presentations over and over again. That would be awesome for all you. And I'd also like to thank Crystal Michael for being great help for anything we ever needed and always being there to talk us through things and help edit. And I'd also like to thank Karen Olmert for editing my papers when she has plenty of other things to do. And we like to thank Laura as well for being an amazing FSI director and making everything so fun and manageable and having a great summer. And we'd also like to thank the University of Northern Colorado for letting FSI stay here and have a great time at FSI this summer. Happy answering your questions now. Okay.
So we said a subcell or instead of non-dividing cells. Does that mean they're the same cells throughout the entire cell?
Not necessarily. What can happen is the hub cells can be replaced, like if they begin to die or if they just get old and the signal stop working. What can actually happen is cells that were starting to differentiate can actually revert, or the germline or the stem cells can actually kind of turn into hub cells. And it's very interesting because the cells know when they need to be replaced, but it's not like, it's not like more hub cells come. So as many die, as many are replaced. But it doesn't happen very often, only if it's needed. So, is there a special reason why you use just the green fluorescent? A special reason? Or is it just like a different protein? Well, it's easiest because it's a fluorescent protein. And like as you see, like we actually made GAL4 like after we wanted to see, but we used GFP to see GAL4 because GFP is easy to stain for. Any other questions? Tony?
So, you may not know this, but is it possible that the Cyst cells, stem cells, are producing a different ligand? Or do we know that this is BTP and it's not common?
No, it is possible that there are different chemical signals being sent. We're not quite sure. We just know that there has to be some connection between Cyst stem cells and germline stem cells because the stem cells are communicating. We know that, but we're not sure if it is a different ligand or a different pathway altogether. But we're testing to see if that ligand could be. Thank you.